Welcome to Shaping Science, a collection of videos from the voices that are shaping science today. I'm Nikki Spahich from The Scientist, and today we are joined by Dr. Verena Mertes, the Director of Biotech 360 at LabVantage Solutions. From groundbreaking discoveries to real-world applications, we'll dive into the ideas and innovations driving the future of science. Today, we'll explore how to turn fragmented scientific data into connected research intelligence with a platform that enables collaboration, insight generation, and the discovery of relationships that would otherwise stay hidden in silos. So, without further ado, let's get into the science. Hi, Verena. Thanks so much for joining me today.
Yeah, hello. Thanks for having me today.
My first question is, in life science research and development, what are the primary challenges and pain points that prevent teams from effectively transitioning from experimental data collection to making actionable decisions?
Yeah, one of the biggest challenges we see actually is data fragmentation. So, scientific data is often scattered across electronic lab notebooks, LIMS systems, spreadsheets, instrument files, and other siloed internal systems. But it's really not just internal data. Researchers are also pulling in data from external sources like public databases or literature, and they may use different formats, different standards, different identifiers. And that really makes it incredibly hard for teams to get a complete view of their experiments, let alone connect insights across disciplines or between in-house and public data sets. And another major pain point we see is the lack of context. So even when data is technically available, it's often not interpretable or reusable. So scientists spend a lot of time searching, redoing work, or trying to trace decisions that were made months ago or even years ago. And then when team members leave, critical institutional knowledge often just disappears with them. So the transition from data to decision it's really not just about data access, it's about meaning, traceability, and also the ability to connect the dots across the R&D lifecycle, including integrating external scientific knowledge with internal experimental data.
And how can semantic technologies help scientists make sense of complex data and effectively share their findings?
So semantic technologies are really a game changer in life science R&D because they help make sense of complex scattered data. So instead of relying on basic keyword searches, semantic technologies or semantic search understands the meaning behind search terms. So how scientific concepts relate, what synonyms exist, and how information connects across domains. For example, let's take HER2. HER2 is a protein that is involved in cell signaling, and it is a key drug target in certain types of breast cancer. But HER2 can also appear as ERBB2 or CD340 in literature and databases. So when you use a traditional keyword search, that might miss these variations because it looks for exact text matches. So unless you search for every possible synonym, you could miss critical information. Now, semantic search, which is powered by ontologies, solves this problem by recognizing that HER2, ERBB2, and CD340 all refer to the same underlying biological entity, and it can pull in all relevant results, even from unstructured sources. So semantic technologies builds on knowledge graphs, which makes it easier to trace data, link insights across studies, and also share findings in a very consistent and structured way. And that, of course, boosts collaboration, supports reproducibility, and helps team to reuse knowledge instead of starting from scratch and reinventing the wheel. Semantic technologies also enable explainable AI. So, since the data is semantically linked and traceable through ontology, the AI output becomes explainable. So, showing which sources and relationships, for example, led to a given answer or recommendation. And that is absolutely critical in life sciences, where decisions need to be scientifically grounded, transparent, and also auditable. So, overall, semantic technologies help researchers move from fragmented, siloed data to a more connected, meaningful understanding, and that lays a strong foundation not just for better science, but also for trustworthy AI-driven innovation.
What significant challenges in data analysis and sharing did your team identify that led you to develop a new platform? And how did you design your solution to address those specific problems?
Yeah, Biotech 360 was born out of a clear gap we saw in the field. So many organizations had already invested in ELNs, in LIMS, which are excellent at capturing and managing experimental data. But in many R&D environments, data is also scattered across a patchwork of systems, from instruments to cloning and construct design tools to inventory management platforms, spreadsheets, data warehouses, and public databases. And these systems don't naturally talk to each other. So without that connectivity, it's really hard to get a complete picture. And AI initiatives often fail because the data is not harmonized or contextualized enough to drive meaningful analysis. So we designed Biotech 360 as a semantic layer that works across existing systems. So it does not necessarily have to replace them, but it can enhance them. And by unifying experimental data like assay results, plasmid sequences, or cell line information with reference data from public sources like UniProt or Campbell, and also enriching information with ontologies, we provide both structure and context. And yeah, together this forms a fair, compliant knowledge graph. That means the data becomes findable, accessible, interoperable, and reusable, giving scientists a way to search, explore, and connect the insights seamlessly. Yeah, but with Biotech 360, we did not stop at the data layer. We also built applications on top of that semantic layer, like our antibody app, our plasmid app, and our semantic search module. And those apps are tailored to life science workflows. And all these apps, they are not just isolated tools, they are really interconnected through the knowledge graph. So insights that are captured in one application can be immediately leveraged in another application as well. So that means teams can go from molecules to decisions much faster with full traceability and also in a format that's ready for AI and automation.
How can breaking down data silos and effectively utilizing artificial intelligence enhance and speed up research and development?
Yeah, breaking down silos unlocks connections that were previously invisible. So suddenly, a compound in one registry can be linked to an assay result in another system, or a plasmid design can be connected to a clinical candidate. So that kind of connectivity accelerates decision making, reduces the duplication, and helps teams reuse knowledge instead of reinventing it. And once all the data is structured and contextualized, AI becomes truly impactful. So you can train models on clean, interoperable data sets. You can also ask natural language questions and get evidence based answers. And you can also go further, like predicting outcomes, identifying novel targets, or optimizing experiment design. So it's not just about doing things faster, it's also about doing them smarter and more effectively. And that's what Biotech 360 was designed to enable.
In which areas of research do you envision this platform will have the greatest impact?
Well, I see the greatest impact of Biotech 360 in precision and personalized medicine, where the success really depends on integrating highly fragmented data sets like genomics, proteomics, clinical records, biomarkers, and making them traceable across the patient journey. And by harmonizing that data, we enable, for example, smarter patient stratification, outcome prediction, and ultimately more targeted therapies. But beyond that, the platform is also incredibly valuable, for example, in biologics development, where teams need to connect, for example, antibody sequences, plasmid constructs, assay results, and regulatory information in a way that's both fast and compliant. I think it also plays a big role in multi-omics research, where combining genomics, proteomics, metabolomics data can only deliver insights if the data is harmonized and searchable across systems. And of course, AI driven discovery is a natural fit by structuring data semantically, making that data fair compliant. We lay the foundation for predictive modeling, intelligent search, and scalable AI. I think it also has a significant impact in more traditional, but still very critical areas like small molecule research, translational research, and clinical development, and even biomanufacturing, where traceability and compliance are really central. So, whether it's cutting edge areas like precision medicine and AI driven discovery, or core R&D functions like biologics development, small molecules, and manufacturing, Biotech 360 can help teams move faster, collaborate better, and make smarter decisions.
And finally, what excites you most about where life sciences research and development is heading in the next 5 years?
I think we are really at a turning point. So, for years, life sciences has been focused on digitizing data, but now we are moving toward making that data truly intelligent. So, this convergence of semantic technologies, AI, and domain specific application means we can finally shift from just capturing the data to actually generating actionable insights. And yeah, what excites me most is a future where scientists spend less time searching for information and more time discovering new knowledge. So, a future where AI is not just a buzzword, but a practical, explainable tool that is really embedded into everyday workflows, and where collaboration across different disciplines becomes really seamless because the data itself is connected, interoperable, and meaningful. And yeah, that's the future we built Biotech 364, and I'm really excited to help shape it.
Thank you, Verena, for joining us today. To learn more about bridging the gap between traditional digital tools and actual scientific insights, please click the link in the description. Thank you for watching. Together, we are shaping science.